Hi, and welcome to another episode of Tom Kennedy's Science. And I'm your host, Dr. Tom Kennedy. Now, today is part 4 of my series on cells. And what we're going to focus on are the eukaryotic cells, and especially the endomembrane system. All those different organelles that all work together and the breakdown and manufacturing of molecules inside the cell. Now, let's get into the endomembrane system. Now, this is a set of internal membranes that divide the functions of the cell into different parts, but it still functions as a single unit. I know, that's kind of weird. It still functions as a single unit. In the endomembrane system, they can either be connected to each other directly or indirectly. And to be clear, the energy converting organelles of the mitochondria and the chloroplast are not part of the endomembrane system. So basically the parts of the endomembrane system, you've got the nuclear envelope, you've got the endoplasmic reticulum, both smooth and rough, you've got the Golgi apparatus, you've got vacuoles, lysosomes, and various transport vesicles. And most people consider that to be part of the endomembrane system along with the actual cell membrane, because all of these things are working together and actually connected to each other both directly or indirectly through a transport vesicle. So the nucleus, this is the brain of the cell, right? And the nucleus houses the DNA, and it's surrounded by a nuclear envelope. Now, you probably have heard that it's a double membrane system. Double membrane. Why in the world would you have a double membrane organelle? Well, it turns out that if you think about the nuclear envelope as a series of flattened stacks stitched together, during cell division, you have to take all the DNA and condense it down into visible chromosomes and then pull them apart. Well, if you're surrounded by a membrane, it makes it hard to pull them apart. So the membrane can easily be disassociated by unzipping it. And then when the cell divides, you can put your nuclear envelope back together easily when the cell finishes with cell division. So that's why it's a series of flattened sacs that resemble a double membrane. It's kind of cool. And to let materials in and out of the nuclear envelope are the nuclear pore complexes. These are large molecular complexes that act as the gatekeeper between the nuclear envelope and the cytoplasm. And there's also the nuclear lamina. This is made up of intermediate filaments that make the nuclear envelope stronger. Now, here's an interesting thing. You have like 17 trillion cells, right? And inside of each one of your cells, you have 43 chromosomes, a little over 3 billion base pairs of DNA. Now, what that means is you have about 6 feet of DNA crammed into every single cell nucleus in your body. That's pretty amazing, isn't it? I mean, 6 feet, right? 6 feet of these things. But the DNA forms this loose structure of both DNA and the proteins that service the DNA called the chromatin. And inside the nucleus, the DNA is highly organized. And depending on which kind of cell it is, the DNA, the same sequences of DNA, the same chromosomes are always in the exact same place in every one of those types of cells. That's amazing. So you got 3 billion base pairs of DNA, and it is highly, highly organized. Another part of the cell nucleus is the nucleolus. And the nucleolus, they will make what is called ribosomal RNA, and the ribosomal RNA is then assembled with different proteins to make the ribosomes. So ribosomes, a small and large subunit, are actually made inside the nucleus and exported outside the nuclear pores. Now, attached to the nuclear envelope is the endoplasmic reticulum, and there are 2 different types, smooth and rough. I wonder how they got their name. The rough ER is rough because it has ribosomes attached to it. Ribosomes make proteins are attached to the endoplasmic reticulum. I wonder what's happening here. Oh yeah, must be making proteins, right? If you thought that, you're right. So what happens here is that you take your ribosomes and they're going to synthesize a protein into the lumen, which is the inside of the endoplasmic reticulum. And inside the endoplasmic reticulum, there's all kinds of proteins in there called chaperones, or chaperonins actually, that help fold these proteins up. And so the rough endoplasmic reticulum is involved with protein synthesis. Now the smooth ER, smooth, doesn't have any ribosomes, not involved with protein synthesis. but it does have three different functions. The first function of the smooth ER is to synthesize lipids like your sterols, cholesterol, right? Estrogen, testosterone. It's also used to help make new membranes. Secondly, the smooth ER is used to detoxify different chemicals in our body, like alcohol. And then thirdly, they store calcium ions. The smooth ER stores calcium ions. And specifically, that is called the sarcoplasmic reticulum, and it's very prevalent in your muscles because you need calcium as part of the process to make your muscles contract. And that's something you'll learn about in plant and animal form and function, which I often teach in the summertime. Okay, let's go back here. We got another structure that's not directly attached to the endoplasmic reticulum, and this is called the Golgi apparatus. I just call it the Golgi bodies. Now, the Golgi bodies are these flattened sacs. They look like pancakes, and each of these membranous sacs is called a cisternae. Now, what they do is they're involved with processing proteins. So if you're making a protein inside the rough ER, it's going to go to the tips of it. It's going to form a transport vesicle where it's going to be taken to the cis side of a cisternae in the Golgi apparatus. And then from there, that protein will get modified. It might be folded. It might be activated. It might be tagged with some type of oligosaccharide. Oligosaccharide. Oligo means small or short. Saccharide means sugar. You put these little small sugars on there that serve as tags to where these proteins need to go. And then eventually, it will exit the trans side of the Golgi apparatus. And because it's tagged, it will go to some place in the cell, like to another organelle or to the cell membrane, or even be exported outside the cell. And I'll talk about that with eating pizza, how that all works together. Wait, eating pizza and endomembrane system? What do they have to do with each other? You'll find out next video. Okay, now in plants, we have vacuoles. And vacuoles, we typically think of them as being used for storage, but they can be quite variable. Like I said earlier, some vacuoles are similar to lysosomes in the fact that they actually carry digestive enzymes. But for a lot of plants, they store ions and water. And in fact, plants can regulate how much water is in their vacuole by moving ions like potassium ions either in or out. And if they're pumping ions in, then water will, through osmosis, move into the vacuole and it will swell. Some vacuoles have lots of proteins in them that are used in seeds for growth. I like flowers. Flowers are really pretty, right? And if you ever notice, flowers have all these pigments that make them colorful. Well, where do you store the pigments? The vacuoles. And, man, some plants are poisonous. They store those poisons in the vacuoles. So as you can see, vacuoles are really, really versatile. And there's two reasons why plants will move water into the vacuoles. Now remember, they're not moving water directly. They're changing the ion concentration in there. They're lowering water potential by pumping potassium ions into the vacuole. And of course, water goes from high to low water potential, so it will move into the vacuole and swell. And there's two reasons why the plant will do this. One is it creates trigger pressure, so it creates a support to the leaves so they don't wilt. And then secondly, have you ever noticed how fast plants grow? Yeah, really fast, right? And one of the ways they can do this, a leaf can expand so quickly, or even a tip of a stem, is that the vacuoles will fill with water and push the cell wall outward. And that's something that we'll learn also in plant and animal form and function. And then we have the lysosome. Now, the lysosomes are found in animal cells. And they're important for like intracellular digestion. So this includes both endocytosis and autophagy. Oh, big words. We'll get back to those. But basically, these lysosomes can carry over 60 different digestive enzymes that you don't want floating around in your cytoplasm, because if they would break down all your lipids and proteins and even DNA in there. So you house them inside the lysosomes. Now, let's get back to these words of endocytosis and phagocytosis. Cells, eukaryotic cells anyways, can bring materials into them. And one of those processes is called phagocytosis, where they can bring large objects like cellular debris from other eukaryotes, bacteria, and other things. And they move the membrane around the object, pinch it off and form a vacuole, and then the lysosome will go up and merge to the vacuole and release the digestive enzymes in there. And they also do what is called autophagy. Auto means self, phagy means feeding. self-eating. Whoa. Yes, we have all of these organelles, and if you remember, mitochondria last about 10 days. Well, why they only last for 10 days? Because they get digested and eaten by the lysosomes. It's called autophagy. So this is actually really important, lysosomes. Think about it this way. You ever seen a young kid, like a 5-year-old or a puppy, full of energy? You ever seen an old dog or grandparents? The kids are running all around, and the grandparents are sitting in the chair watching them run around. As we age, we tend to lose energy. Well, what gives us our energy? Mitochondria, right? They're making all of our ATP. Well, as we age, the rate of autophagy slows down. So we begin to accumulate these older, less efficient mitochondria. That means we make less ATP. That means we have less energy. That means we move less. We exercise less. Our muscles get smaller. We atrophy. We move less. You see the self-feedback here. We got a positive feedback loop, and that's part of the aging. So one of the areas of research right now is learning how to increase the rate of autophagy or reset it back to a more youthful state, like in your 20s. Wait, you guys are mostly in your 20s. I'm not. I'm working very hard to keep my rates of autophagy high by doing lots of exercise. But if we could actually reset that, then we could start resetting the ages of our cells and slow down the aging process. So this is another reason why we study lysosomes in a process like autophagy, because it's part of the puzzle to living a longer life. And then also, a lot of diseases associated with aging, like Alzheimer's and Parkinson's and cardiovascular disease, are also in part caused by changes in the rates of autophagy due to our lysosomes. Pretty wild, isn't it? Okay, the cell membrane is also considered part of the endomembrane system because it's the endomembrane system that makes new cell membranes and also makes all of the proteins that are embedded or exist on the cell membrane. Now, think of it this way. Cell membranes are incredibly important to cells because cells are a system, a set of interacting components defined by a boundary, and that boundary is set by our cell membrane. And our cell membranes have selective permeability. That means only certain things can enter or exit a cell. So if you're oxygen, carbon dioxide, even water, because water is a small molecule, they can readily pass through cell membranes without the cell doing any work. But cell membranes are incredibly complex. Not only do they have all types of signaling molecules and receptor molecules, they also have all types of transports and pumps embedded in that membrane. So the cell can regulate what comes in and out of the cell to maintain homeostasis. And one thing that's interesting about cells, they are not in equilibrium with their environment, right? They maintain a high degree of order inside the cell, and it's that membrane that helps maintain homeostasis and create this different environment inside the cell. Well, I hope you learned a little bit about the endomembrane system and other various parts of the cell in this lecture. Till next time.